Good evening, Prof. Azhar. The topic of my research proposal is the investigation of the propagation of virulent Newcastle disease in animal cells grown on newly developed ultraviolet or ozone-treated polystyrene microcarriers. The overview of my presentation includes background of study, problem statement, research objectives, research methodology, expected result, conclusion, and finally, references. First, I'll move on to the background of study. What is Newcastle disease? Newcastle disease is categorized by the Office International des Epizootiques as a list A transmittable disease of poultry. It affects most species of birds such as chickens, turkeys, pigeons, parrots, ducks, geese, and quails. Chickens are the most vulnerable host towards this Newcastle disease. Newcastle disease is transmitted through droppings and secretions from the nose, mouth, and eyes of infected birds, and also through contaminated water, feed, and transport. The first outbreak of the epidemic was in Java, Indonesia in the year 1926, and later in Newcastle upon Tyne, England in the year 1927. However, there were prior reports regarding the earlier outbreaks in Central Europe, which was in 1896, that caused death of all chickens in Western Isles of Scotland. After that, in the year 2002 to 2003 in California, there was also depopulation of almost 4 million birds in 2,662 premises due to this disease, which had resulted in $200 million worth of losses. Currently, the Newcastle disease is prevalent throughout the world, including Central and South America, Asia, Middle East, and also in Africa. Newcastle disease is caused by Newcastle disease virus, which is also known as avian paramyxovirus 1, which belongs to the genes of Avula virus under the family of Paramyxoviridae, classified under the order of Monomygavirales. It often appears in spherical shape with diameters around 100 to 500 nanometers. Apart from that, it also has a non-segmented, single-stranded RNA genome of negative polarity. Genome length of about 15,186 nucleotides. The genome also comprises of 6 genes, which encodes the proteins in the order of 3 to 5. This is basically the microscopic structure of a Newcastle disease virus. This is the RNA strand. The N represents the nucleoplasmic protein, P represents the phosphoprotein, L is the large polymerase, and M is basically the matrix protein. And on the outer region, E is the envelope, HN is the hemagglutinin neuraminidase protein, and finally F is the fusion protein. Strains of Newcastle disease virus are basically classified into 3 groups according to their pathogenicity. The first group is the lethogenic group, which has low virulence that causes only clinically mild or unapparent respiratory disease. The second group includes the mesogenic group, which is of moderate virulence, and it causes respiratory and nervous signs with moderate mortality. And the next group includes the velogenic group, which is of high virulence that of course causes 100% mortality, and it is further divided into 2 groups, which is the viscerotropic velogenic group that produces lethal hemographic lesions in the digestive tract. And as for the neurotropic velogenic virus strain, it produces neurological and respiratory disorders. Newcastle disease vaccines are further divided into 4 groups. Firstly is the live lethogenic vaccines, which is derived from virus strains that have low pathogenicity. It is caused by the La Sota, Hitchner B1F, and also V4 strains. It produces adequate immune response. And apart from that, the optimum doses of this type of vaccine is between 10 to the power of 6.5 to 10 to the power of 7 of embryo infected dose 50 per bird. The second type of vaccine is the live mesogenic vaccine, which is derived from moderate virulent strains caused by the Muktaswar, Komarov, Rocaine, and Hertfordshire viruses. It is used as a secondary vaccines after a primary vaccination with a lethogenic vaccine. The optimum dose for the vaccine is approximately 10 to the power of 5 of embryo infected dose 50 per bird. The third type of Newcastle disease vaccine is the inactivated vaccines. As for this vaccine, the allantoic fluid containing Newcastle disease virus is treated with inactivating agent such as formalin or beta-proteolactone. An adjuvant such as mineral oil is usually then added to make the inactivated virus more immunogenic. The inactivated vaccine against polio disease is one type of this inactivated vaccine. Lastly, another type of recombinant vaccine, which comes under the Newcastle disease virus vaccine, is part of the genetic material of virus that codes for a surface antigen, is taken and inserted this into another type of virus. So the fusion gene inserted into herpes virus of the case is used to produce Newcastle disease vaccines. Virus culture. A virus is a biological agent that replicates inside the cells of living hosts. The main purposes of viral cultivation is to isolate and identify viruses in clinical specimens, to do detailed research on viral structure, multiplication cycles, genetics, and effects on host cells, and lastly is to prepare viruses for vaccines. So there are two types of virus cultivation system. Firstly is the embryonated egg systems, and secondly is the mammalian cell culture system. What is embryonated egg system? So embryonated egg system was basically discovered by Ernest William Pasteur in 1931. It is a traditional method for producing viral vaccines. Embryonated egg offers a variety of sites for the cultivation of viruses such as the chorioallantoic membrane, amniotic cavity, allantoic cavity, yolk sac, embryo, and air sac. As you can see here, the virus can be injected through various cavities mentioned here. So the optimal age of egg for virus inoculation is 9 to 11 days of incubation. And the eggs are incubated after inoculation at 35 to 37 degrees Celsius for 4 to 7 days and candled daily to check vitality. Death of the embryo is slower if inoculation is via the allantoic cavity, which is this part of the egg. So the inoculation via the allantoic cavity can result in high virus titer. Mammalian cell culture is another type of cell culture used for virus cultivation. So it is the major field in modern biotechnology today. Cell cultures have replaced embryonated eggs and for virus production. It is more convenient and the preferred type of growth medium for many viruses. In the year 1949, Ender and his colleagues proved that poliovirus could replicate in a cell culture medium. Cells of a mammalian derived from specific tissues cultivated and reproduced in an artificial medium. Products from the cells include viral vaccines, monoclonal antibodies, interferons, and recombinant therapeutic proteins. Production of viral vaccines was one of the earliest purposes of cell culture development. Animal cell lines used for production of viral vaccines include Vero, Mbbk, MSKK, CEF or DF1, and also BHK. So Vero, Mbbk, and DF1 cell lines will be used for this experiment in this project. So what is serum? Serum is basically the common supplement in cell cultures. It is an extremely complex mixture of plasma protein, polypeptide, fat, carbohydrate, growth factors, and also hormones. It is obtained from the bovine fetuses of cow. For example, the fetal bovine serum, horse serum, and also fetal calf serum. Another type of growth medium that can be used for cell culturing is the serum-free medium, which is developed by White for the first time in the year 1946. So it contains no animal serum, but it only contains serum constituents or substitutes such as discrete proteins or bulk protein fractions or plant extracts. Okay, the mammalian cell lines include Vero cell line, DF1 cell line, and Mbbk cell line. So what is Vero cell line? Vero cell line is derived from the kidney of an African green monkey in the year 1960s. The cell line was founded by Y. Yasamura and Y. Kawakita at the Kaiba University in Japan on March 27, 1962. It is a continuous and anchorage-dependent cell line. It can be readily adapted for growth in bioreactors on microcarriers and provide consistently higher yield of viruses. It also provides greater vaccine purity as it contains less contaminating cell debris. Secondly is the DF1 cell line, which is an immortalized continuous cell line of chicken embryo fibroblasts. It is anchorage dependent and it is derived from 10-day-old East Lansing line eggs. It was named after its founder Douglas N. Foster. Thirdly is the Mbbk cell line, which is the Medin-Darby bovine kidney cell line, which is an epithelial cell line. So it is derived from the kidney of an apparently normal adult Bos taurus in February 18, 1957 by S.H. Medin and N.B. Darby. Scaling up of animal cell culture is basically to obtain high quantity of cells and for getting a relatively high number of cell products such as viruses, monoclonal antibodies, interferons, or recombinant therapeutic proteins. It is basically for metabolite or enzyme extraction. So the scaling up of anchorage-dependent cells can be done in spinner vessels, roller bottles, hollow fiber system, glass bead reactors, and also by microcarrier system. The cells which grow in suspension are scaled up in stirred tank bioreactor or air leaf fermenters. So the factors affecting cell growth in bioreactors include agitation, aeration, pH, oxygen, temperature, cell inoculum, and also microcarrier concentration. So basically the type of scaling up system which will be used for this project is the spinner vessel and microcarrier system. And the factors that will be manipulated for the cell growth includes agitation, cell inoculum, and also microcarrier concentration. Bioreactors for anchorage-dependent cells. As you can see here, this is the spinner vessel. So this is they can be either plastic or glass bottles, and it contains a suspended central Teflon pedal with a central magnetic stirrer shaft, which turns and agitates the culture when placed on a magnetic stirrer. So the side arms for the addition and removal of cells and medium and gassing with carbon dioxide enriched air. It is designed up to a capacity of 10 liters, and the stirring is stable at a rotation speed of between 10 to 300 RPM. It is usually siliconized by applying dimethyl dichlorosilane to prevent attachment of cells to the inner glass surface. Microcarrier system. So what is microcarriers? Microcarriers are small particles made of cellulose, dextran, glass, polystyrene, polyacrylamide, sephadex, collagen, or gelatin. So it has a spherical shape and presents a surface structure and composition that promotes cell addition and growth. The characteristics required by microcarriers include it has to be small to maximize the available surface area for cell growth. It also have to be light to allow easy suspension in culture medium, transparent to allow easy observation of cell attachment and growth. Besides that, it must also be charged to allow cell attachment onto the surface. So the first culture of cells on microcarriers was developed by one vessel in the year 1960s using DEAE Sephadex PM50 gel beads. And the first product produced was an inactivated polio vaccine. So this is a new type of microcarriers, which is the ultraviolet or ozone-treated polystyrene microcarriers. So it is a new microcarrier which was developed by Arifin in the year 2016 using methods that are relatively easy, simple, and very low cost. So the ultraviolet or ozone-treated polystyrene microspheres have shown to improve hydrophilicity or surface wettability of the microspheres, which is essential for good cell attachment and proliferation of cells. So it had resulted in a high density of cells compared to the use of other commercial microcarriers. It could also serve as a low-cost alternative to commercialized microcarriers available in the market today. So this is basically the picture of the ultraviolet or ozone-treated polystyrene microcarriers with the cells attached to them. So the detection of viruses can be done using various techniques, which can be classified into 4 categories, which is the detection of virons, detection of infectivity using cell cultures, detection of virus nucleic acids, and also the detection of virus antigens. So basically the detection method used for viruses in my project is the detection of infectivity using cell cultures and also detection of virus nucleic acids. And apart from that, the physical assay that can be used is the hemagglutination assay. And also for the infectivity assay, there are 2 types, which is the quantitative assay that includes viral plague assay and the quantal assay, which includes Tcid50. Detection of infectivity using cell cultures. So the cytopathic effect refers to pronounced morphologic changes induced in individual cells or groups of cells by virus infection, which are easily recognizable under a light microscope. So the cytopathic effect includes rounding of the infected cell, shrinkage, increased refractivity, fusion with adjacent cells to form syncytia, and also the appearance of nuclear or cytoplasmic inclusion bodies. As for the next method, the detection of virus nucleic acids. So this is the probability of detection of virus nucleic acid that can be increased by amplifying virus DNA using polymerase chain reaction, also known as PCR. RNA can be copied to DNA and amplified using a reverse transcriptase PCR, known as RT-PCR. So it is an amplified product that can be detected by electrophoresis in an agarose gel. So what is hemagglutination assay? So it is a physical assay which determines the quantity of virus, unlike their infectious or not. So the sufficient amount of virus causes agglutination of red blood cells. The red blood cells that are agglutinated evenly coat the bottom of the well. The minimum amount of virus needed to cause agglutination is determined based on serial dilution. So basically in this picture, there are different types of sample available with different amount of dilutions. So the virus titer is basically measured in HA units per ml. Next is the Tcid50 method. It is a quantal assay to find out if an inoculated cell culture either develops a cytopathic effect or not. So the tissue culture infectious dose, defined as the dilution of virus, is required to infect 50% of the cell monolayers. So the Tcid50 determination can be used to estimate a virus titer in TFU or vice versa using this formula, which is 1 Tcid50 per ml equivalent to 0.7 TFU per ml. So the other methods used to estimate the endpoints include Reed-Muench and Spearman-Haber method. I'll move on to the problem statement of this research project. So the Newcastle disease is the most prevalent and contagious bird disease, which is an endless threat to our poultry industry. So it has prompted a massive economic losses, and the only way of preventing this disease is through vaccination. So the traditional method for vaccine production used was by growing virus strains in embryonated chicken eggs. However, the traditional method has many drawbacks, such as poor quality, high labor intensity, time consuming, needs high amount of specific pathogen-free eggs, and also requires big area for incubation of eggs. Therefore, the production of Newcastle disease vaccines using animal cell culture technology is one of the methods that has high possibility to overcome the disadvantages in using embryonated chicken eggs. Animal cell culture technology has many advantages since the method is rapid, convenient, less expensive than eggs, supports easy scale up, and it also allows evidence of viral proliferation to be examined microscopically. However, the use of serum in cell culture medium has many disadvantages such as high physiological variability, high cost, high batch-to-batch variability, and risk of contamination with virus, prions, and also microplasmas. Apart from that, serum obtained from bovine fetus of cow also raises ethical and animal welfare problems. Therefore, application of animal cell culture technology using microcarriers in serum-free cultures is able to produce Newcastle disease vaccines of higher quantity and purity. Next, I'll move on to the research objectives of my project. Firstly is to adapt 3 cell lines such as Vero, DF1 and Mbbk cell lines in virus production serum-free medium and determine the suitable period for virus cultivation for each one. So the second research objective is basically to investigate the propagation of Newcastle disease virus strains such as La, Sato, and Mukteswar strains in virus production serum-free medium adapted cell lines and determine the cell line which produces highest virus titer and infectivity. The third objective is to upscale the cell culture in spinner vessel by optimizing the process conditions such as the cell inoculum, microcarrier concentration, and stirring speed. The last objective is to propagate the Newcastle disease virus in selected host cell line using optimized culture conditions. Research methodology. So the first step is basically to construct growth profiles of 3 different cell lines such as the Vero, DF1 and Mbbk cell line in serum supplemented medium. Secondly is the adaptation of the 3 cell lines of in virus production serum-free medium using T-flask. The next step is to determine the cell density, viability, glucose uptake, and lactate production of the 3 cell lines in each stage of adaptation. The fourth step is to infect the 3 cell lines with 2 types of Newcastle disease virus, such as the La, Sato, and Mukteswar virus strains. The fifth step is to determine the cell line with highest virus titer and infectivity using Hemagglutination assay and Tcid50 assay. The sixth step is screening and optimization of process conditions such as stirring speed, cell inoculum, and microcarrier concentration in spinner vessel for maximum cell density. Next is the production of Newcastle disease virus in spinner vessel, and finally is the confirmation of virus sample by using reverse transcriptase polymerase chain reaction, which is also known as RT-PCR. Okay, the expected result of my research project is that the highest virus titer can be achieved by cell culture using ultraviolet or ozone-treated polystyrene microcarriers in spinner vessel. From this, I would like to conclude that the process developed might be ready for live Newcastle disease vaccine production, and perhaps it can also serve as a platform for producing vaccines for other diseases. So that's all from me. Thank you, Prof. Azhar.